So, my name is Arjan Palstra. I'm a senior researcher and a fish physiologist at Wageningen University Research, and I will present for you a study that we did in collaboration with Ana Roque from ERTA, which is on the optimal flow for seabream during land-based rearing and the activity patterns in the sea cage. So first I will start off shortly with the objectives of the study. And basically it's a study on two topics. First one is during land-based rearing to study optimal flow regimes. And secondly, how do seabream behave in the sea cage? So the activity patterns over the day and over the weeks. Finally, I will end with conclusions and recommendations and also some perspectives for the near future. So basically this study represented subtask 2.2.2, which is the effects of optimal water current inducing swimming behavior, which was part of task 2.2, the assessment of rearing conditions to improve juvenile quality and fish welfare within work package 2 of the MED-A project. So we led this subtask and we collaborated with ERTA in order to design exercise protocols for seabream in RAS to increase robustness and to evaluate the effects of these protocols during on-growing by monitoring behavior with accelerometers. Basically, this objective comes from two sides. First of all, we have performed a lot of studies on the benefits of exercise, swimming different species. Among others, studies were published in this book in 2013. And the other entrance was a workshop that we organized in 2015 at the Aquaculture Congress together with industry, where we investigated the benefits of a prolonged on-land rearing period. Together, they made this objective within the MED-A project. So let's move to topic A, optimal flow regimes. What we did is we had a setup of 3 of these 1500 liter tanks. And you can see in the middle, you can see an inner ring that made that the flow was quite uniform. You can see that we also lowered the water level in order to increase water flow. And you can see a fish swimming there. We tested 3 regimes, which were created by the water inlet, basically a minimal flow treatment. Let's call it 0 body length per second, and then swimming at 1 or 2 body length per second. Of course, because fish were growing during the experimental period, every time we increased the flow corresponding to the speed, and we did that over the full period of 8 months. By doing this, we were able to reach maximal flows between 40 and 50 centimeters per second. So that corresponded to 2 body lengths per second of the biggest fish at the end of the experimental period. For each of these tanks, we had about 1,100 fish coming from Kulmar, still quite small, with a body weight of 2 grams and a length of 4 centimeters. These fish were manually fed twice per day ad libitum, and every 2 weeks we measured length and body weight, and after 3 months they were big enough to tag with pit tags. At the end of the experimental period, we performed a stress challenge and measured cortisol. We did extensive measuring and sampling, biometrical measurements. We weighed the intestine, liver, intestinal fat, spleen, and heart. We also took samples of liver, white muscle, heart, and blood in order to measure plasma glucose, lactate, triglycerides, cholesterol, growth hormone, and IGF-1. And we did gene expression on heart and white muscle growth hormone receptors and IGF-1 receptors. And we performed RNA sequencing of the white muscle transcriptome. Also, we selected fish of this group for the next stage, so for following fish in the sea cage by accelerometry. Move to the results. Here you can see on the x-axis the different time points where we measured both length on the left side and weight on the right side. You can see a significant difference, which are particularly expressed by body weight. So basically, after about 4 months, you can see that there is a significant difference for the fish swimming at 1 body length per second and at 2 body lengths per second. And I'm trying to take this away so that I can see my own slide. So we see that at 1 body length per second, when the fish were about 20 grams, the biggest ones, they increased about 28% in body weight. The 2 body length per second, that was 40%. And at the end of the experimental period, so after 8 months, we see still that that fish at 1 body length per second were 15% heavier, and at 2 body lengths per second, 36% heavier. And also importantly, there was also less variation. So you see in both exercise groups, you can see that the variation was about 25% less. So at the end, we performed a stress challenge test with, which was performed by a netting stressor. So we netted the fish for about 3 minutes. And here you can see the result. On the x-axis, we have time, the time after the netting challenge. And on the y-axis, you can see the cortisol levels. We see 3 lines. The light gray are the controls. The darker gray, they are the fish swimming at 1 body length per second. And in black, the fish swimming at 2 body lengths per second. And we see that just before the netting challenge, there is a significantly lower level of cortisol in the fish conditioned at 1 body length per second. No difference between the 2 other treatments. And we also see that close to peaking and already during recovery, that the levels are not significantly different, quite close to each other when peaking and with large individual variation when fish were recovering. Now, this is a whole lot of data. I will guide you through it. We have basically 3 columns. The C is of the control fish, and then we have the fish at 1 body length per second and 2 body length per second. You can see the average and the standard deviation. So first of all, what we see is indeed swimming enhanced growth. So the fish that were swimming, the exercise group, they increased in weight, which was also expressed by an increased condition factor. We also see that they have bigger hearts, so the heart weight was increased. And we see that they had more fat, so mesenteric fat was higher in the fish that swam. And that was a little bit surprising, and I will get back to that, because we would not expect that fish that swam had more mesenteric fat. As for glucose, we see that the levels were increased in fish swimming at 1 body length per second. Now, we have swimming enhanced growth, but while investigating the parameters of the growth axis, we didn't find any evidence of changes in plasma growth hormone and IGF-1, but also no differential expression in their receptors. Now, importantly, we already saw in the control group that there was a level of about 30% of the fish that showed abnormal morphology. So deformities ranging from very few to quite extensive scale, as you can see here on the 2 pictures. And that was already in the control group, a little bit higher in the fish swimming at 1 body length per second, but really significantly higher in the fish swimming at 2 body lengths per second, increased up to 48% of the fish. Now, by investigating gene expression through RNA sequencing, we compared the fish swimming at 1 body length per second with the controls, and the fish at 2 body lengths per second with the controls. We had a number of significantly expressed genes, 87, 65, and 10 in common, which can be considered marker genes for exercise effects that you can see here. Basically, all these differentially expressed genes, they showed 3 major pathways that express the changes. Of course, I would say in muscle development and the molecular regulation of that development, meaning that you see by swimming, you increase the weight of the fish, and that is most extensively having an effect in muscle development. We see genes involved in the muscle contraction. So we hypothesize that these are mainly genes involved in the adaptation to skeletal abnormalities, but that is still with a question mark. And basically, like always with exercise, we see a whole lot of immune genes being differentially expressed. It could be that they play a role in disease prevention, so the relation between exercise and disease prevention by myokines, which is known from mammals particularly. but also known from mammals is the exercise-induced inflammatory response, which is not bad, which is good because it causes a repair, a regeneration, and a development of the muscle. Moving to the second topic, where we try to follow the activity patterns of the seabream in the sea cage. That was performed over a period of 6 weeks in November, December. We transferred 300 fish from the facilities at IRTA to Mallorca, where we had them in an experimental sea cage that you can see here on the picture. 300 fish, and 30 of them we implanted them with an accelerometer. And on the picture on the top right, you can see how we implant these 2 centimeter sensors in the fish. No harm done. As soon as they are back in the tank, they start eating. So with seabream, that's having a low impact. We see that the length was about 18 centimeters, 170 grams. When starting, they were fed once per day in the morning at 11 ad libitum, and at the end of the experimental period, we did biometrical measurements and weighed intestine, liver, intestinal fat, spleen, heart, and fillets. Now, then you're there with about a few 100,000 data points, and you analyze them and you all put them in a single graph, and that is what you see right now. So on the x-axis, we have the hours of the day from 12 to 12. And on the y-axis, we have the activity, but take care, this is acceleration. So you measure accelerations and you take that as a proxy for activity. And what you see is a W-shaped pattern. So basically, seabream are relatively not nocturnal. They wake up at 6. Towards feeding, they show increased activity, food anticipatory behavior. Then feeding at 11, a period of increased activity up to about 2, then activity decreases. You could say some sort of a siesta, and in the afternoon, in around 6, they increase activity again. You can also see a development over the weeks. Well, basically in the first week, there's increased activity, probably because of an habituation effect. And then over the weeks, you can see activity dropping with dropping temperatures. So it's, as you can see, it's a perfect tool, these sensors, if you have some marker fish, to have an insight into the activity and to use these data, for instance, for when to feed the fish, which can be at the start of activity periods. Now let's go back to the original question. If we take the fish, the control fish, and the swimmers at 1 body length per second and 2 body length per second, we see that they all show a similar activity pattern, so no changes there. We do see that the fish swimming at 1 body length per second, they have a significantly higher activity than the other 2. However, it is significant, but quite marginal, and that's why we did not consider these differences further in the analysis. We also wondered, what exactly do we measure? Acceleration is taken as a proxy for overall activity, but in fact you're measuring acceleration. So could it be that we only measure unsustained activity? What happens if these fish, they swim in sustained manner, making their circles in the sea cage? What do you measure then? And we did a study on that, which goes a little bit outside of the MEDAID project, but I think it's interesting to interpret these results. We did a study where we had 4 swim tunnels in which we swam seabream that had a sensor or not. We performed accelerometry. We analyzed the locomotion behavior by using a high-speed camera, and we performed respirometry. And basically what you see, the main result was that when you increase the speed, so fish swim faster, you can see that the accelerations that you measure with the sensor, they go down. So swimming speed increases, fish swim more and more sustained, and you can see that accelerations go down. So basically, this behavior is not really covered in what we measured with the sensor. So proxy for activity, yes, but still only based on unsustained movements. And that brings me to the conclusions, the recommendations. So what we can conclude is that we have an optimal flow regime when you exercise the fish at 1 body length per second. Many benefits. like enhanced growth, more uniform growth, less chronic stress, indications for more robust fish, like having larger hearts and higher condition factors. You can also see changes in energy metabolism, so higher plasma glucose. Fish are more active significantly, but slightly. And you see, you can expect changes in muscle structure, development, repair, and regeneration capacity on basis of the gene expression patterns that we see, although we did not really measure that. Also, importantly, you see that there's a slightly higher level of lordotic abnormalities, and that needs some refining. For instance, by starting the exercise protocols a little bit later in life, so exercise is not such a burden during early development. Or you can also think maybe the fish should not swim constantly, not 24 hours per day, but include rest periods. Make it a working day and swim them 8 hours per day, for instance. But as again, as I said, that needs some refining. We also see that extending the on-land period makes it possible to benefit from these physiological effects more than when you bring them to the cage at 20 grams. We see the enhanced growth. We still don't know if that was because of increased feed intake and or lower FCR. At least we know that you can shorten the production cycle. Importantly, fish had more uniform growth, less variation, a more uniform product. We see seabream may be more robust, and that could lead to lower disease and mortality rates, less ethical concern, and perhaps more societal acceptance. As for muscle structural changes that can be expected, also a topic that needs further exploration, but you may expect more meaty flesh. And of course, it depends on the consumer if that is considered better or not. Exercise seabream may also resemble more wild seabream. So if that is important for whole fish sale, that could certainly be a factor that can be considered. And finally, you can see that there's a lot of sensor development going on for the real-time monitoring of animal management and welfare. And you can see that you can really benefit from applying those sensors in precision farming, integrated networks, and big data strategies. To summarize, swim your fish at 1 body length per second, I would say, needs some more refining, but certainly the benefits are there. If you have further questions, you can ask them now, or you can always email me. Most of this is already published in 2 papers, and a third one is coming, so you can always check those. I would like to thank several people: Pablo Arachuela Lopez, Jaume Perez Sanchez, Ron Dirks, Pauline Jeannet, Leo Kruid, Albert Leon Delgado, and Job Stokkermans, and of course the zootechnicians and research assistants at ERTA, Limia and Medea, and Caris. And of course, I would like to thank you for your attention.